Good morning, all. Welcome back to Chemistry 1406. Uh, we're going to start talking about some proteins and amino acids now in this lecture. It's my favorite part of chemistry, partly because I'm a formally trained biochemist. So I might have a lot of different examples in here from my life for you to glean some insight in this particular topic. In. So let's go ahead and get started. Let me change the view like I always do. There we go. Yes, yes. Okay, so you may have noticed that there's a different PowerPoint presentation background now for the title slide. It's just something I guess that Microsoft is doing this thing called Microsoft Intelligence. And I guess it noticed that I was doing a lot of chemistry lectures, and so it decided, hey, maybe this is a good chemistry lecture background. Interestingly enough, though, I have those exact ball and stick models here. So I guess it's a little creepy, but Microsoft knows me. Anyway, let's go ahead and go on. So what are proteins? Well, proteins are one of the biomolecules that I spoke of before when I told you we were talking about biomolecules. I was talking about when I was talking about that. Then I was talking about those four carbohydrates, lipids, and then proteins. And then I think last time I said there were three. I made a mistake there. There's actually four. The last one is nucleic acids. So the four types of biomolecules that we're studying and are going to study are the lipids, the carbohydrates, the proteins or amino acids, and then the nucleic acids. Right. So we've already done the carbohydrates. We've already done the lipids. Now we're going to do the proteins. And then after the proteins, we'll do the nucleic acids. So what are proteins? Well, proteins are made up of essential amino acids, right? And regular amino acids. They do plenty of things in your body and every other living organism's body. And they are basically 3D models of a bunch of amino acids that have come together, right, to form this big 3D molecule, three-dimensional molecule, right, that performs some type of function, right? Whether that would be a structural function or an enzymatic function, catalysis function. Proteins are what we use to do things with, right? So let's just quickly go through the formal stuff about proteins here, right? They're polymers of amino acids, right? The 20. When I was in grad school, I actually, not just grad school, undergrad too, one of the things we had to do was memorize all 20 amino acids, plus the two extra ones, and then also their first letter, or their single letter designations, their three-letter designations, and a bunch of other stuff. But don't worry, I'm not going to make you memorize all 20 amino acids, plus two. But yes, they are polymers made of the 20 different amino acids. A few of them are essential because your body can't actually make them themselves, right? There's a process in our bodies called amino acid metabolism, and we can make some amino acids ourselves, but most of the amino acids that we need are essential, and we get them from eating things like meat, right? Or soybeans, if that's what you want to do, or drinking in them from milk and stuff. So they different characteristics because the functions of each proteins differ because they're made up of different amino acids, right? Different combinations of amino acids that have different our side chains. Remember what I was talking about before? How not amino acids, but molecules have a center part of them, and then there's an R for the rest of the molecule. Well, it's the same thing with amino acids. So with those amino acids, there's 20 different variations, and then you can get an infinite number of them put together in 20 different variations, and those all will interact differently, providing you with a very diverse set of functions and structural capabilities for proteins, right? And that's delineated here because you can actually make structural components like cartilage, muscle, hair, nails, right? Or they can do the enzymes, which we'll get to in the next lecture. My specialty in biochemistry is actually enzymology, and it's interesting because we're only going to spend one lecture on enzymology or like enzymes, which is like maybe less than an hour on it. But there's an entire class called enzymology and enzyme catalysis, and one of my professors was actually he was the student of the enzyme of sorry, I said that wrong. He was one of my professors when I was in grad school was the student of the father of enzyme kinetics, Mo Cleland, who was up in the University of Wisconsin. Him and Perry Fry and a couple other people at the University of Wisconsin wrote the book on enzyme kinetics, and so I'm very proud to be a part of that pedigree, that lineage from Mo Cleland to my boss at grad school, who is Steve Mansur-Body, and then me. I was actually able to publish a paper with Perry Fry, who's also another one of those biochemistry enzymology moguls too, and I was very proud of that. So anyway, the point is, you can take an entire semester class on enzymology, whole textbooks on enzymology, but we're only going to cover it in one lecture. But that's okay though. I'll give you what you need to know about it. But yes, what are enzymes? Enzymes are responsible for biological catalysis, right? So what they're biological catalysts, which means they speed up reactions, right? Or more specifically, these enzymes are proteins, and they will make it so that the activation energy, if you remember from a previous lecture, they'll make it where the activation energy of a particular biological reaction that's required for you to live doesn't require as much activation energy, so it can actually proceed, right? And we'll see that in a second. And just another example, hemoglobin and myoglobin, they're proteins too, and they transport oxygen in the blood. That was actually, I believe, one of my first examples for you. Hemoglobin and the heme with the iron center, and it will bind oxygen, right? So I believe that was the very first example I ever gave you for enzymes. And it was like maybe the first lecture. Wow, that was a long time ago. So let's just quickly go through some of this stuff here. There are classes of proteins, right? There are structural proteins, which is obvious. They provide structural components like collagen, which is in tendons and cartilage, like in your nose. And then there's keratin, which is hair. It's in your nails. There's also contractile proteins. They make your muscles move, like myosin and actin. Transport proteins, hemoglobin, which is the one that transports oxygen throughout your body, and then lipoproteins, which we were talking about in the last lecture, high density lipoproteins and low density lipoproteins, right? They're those, and then there's storage proteins too, casein for proteins of the milk, and ferritin, right? Which ferritin is like ferrous or ferric, like iron, so it stores iron in the spleen and liver. There are hormones. There are proteins that are hormones, right? One of those is insulin. Insulin is actually a very short. Insulin is a very short. Sorry about that. My dog is in the room with me right now. He's sleeping. His name's Yogi, and he was making some really loud noises, and I'm like, Yogi, you okay? My dog's name is Yogi, if I didn't say it already. So I had to make sure he was okay. He's okay. I think he was having a nightmare or something. Anyway, back to this. Excuse me. So there are hormone proteins too. One of them is insulin, right? Insulin is a very short peptide, which you'll learn what a peptide is. It's just a peptide is a string or a polymer of amino acids, and that's what insulin is. It's a short string of amino acids. And if you recall from our previous lecture before that, insulin is responsible for signaling your body to store glucose, right? And then, of course, we have enzymes, which I already talked about. There are all kinds of enzymes like sucrase, which hydrolyzes sucrose, trypsin, which hydrolyzes proteins, which means breaks them apart using water. And then their protection, which is they recognize and destroy foreign substances like the ones in your immune system, right? Your immune system. Those are like the immunoglobulins, right? Which are just they're antibodies, which are responsible for identifying antigens in your immune system. Um, so I like to actually break this down even more simply into the fact that there are two types of proteins. There are structural proteins, right? And then there are functional proteins, right? And the functional proteins are all enzymes. They do a process, right? They carry out some kind of process. Whereas the structural proteins are the ones that are like, they make the structure in a body or a cell, right? So that's how I like to categorize them. They're either functional proteins or they're structural proteins. Okay. So even for example, you have structural proteins here, the first one, but then you have contractile transport proteins, right? Uh, all that stuff after this, after structural, they're all functions, right? They're all functional proteins. So you have the one structural protein right here, but then all these guys here I would consider functional proteins because they carry out some kind of purpose, right? And then the main functional protein, is the enzyme, right? The enzyme, which we spent a lot of time talking about enzymes. Wow, that was a lot of time spent on this particular slide. Let's keep going. So, I was talking about amino acids in the last slide. What are amino acids? So, basically, amino acid is composed of this thing right here called an alpha carbon. It's also like the central carbon of the molecule, right? And connected to that is one hydrogen, which is right there. It's always there, right? And then on one side is an amino group or ammonium group, right? Which we call the N-terminal side, N for ammonium, right? And then terminal because it's at the end. And then on the other side we have a carboxylate, right? Which we call the C-terminal side, right? Because it's got a C for carbon, right? And then last but not least, because there are 4 bonds to carbon, right? We have the R group. And remember what I said what an R group was? It's the rest of the molecule, right? So for this particular protein here, right? The R group is a methyl group, right? So that yellow thing right there, the yellow part, that methyl group there, the R group is the part of amino acids that's variable, right? I mean, you're only learning about 20 amino acids here, right? I mean, I'm not even sure if I'll go through all 20. No, I will go through all 20. You're only learning about the 20 amino acids here, but there's multiple amino acids. There are alpha amino acids, which are what these are, because everything's coming off of the alpha carbon. There's beta amino acids coming off the second carbon. There's also modifications on the 20 original amino acids too. So there's a lot more than just 20 amino acids. But in this class, we're only going to focus on the 20. And in all those 20, this one right here, the R group, is different on all of them. So here's some examples. We have valine here, right? And valine's R group, right, is this CH, CH3, CH3, right? This alkyl group, right? Whereas on asparagine, the R group is the CH2 and then the amide group, right? This amide right here, right? So those are two different amino acids right there. Valine and every other amino acid has a 3-letter designation. One of them is Val. For valine, it's Val, VAL. It also has a one-letter designation, which is V for valine. We wish that they were all just the first letter of the the first letter of the amino acid's name, but it's not. So here we have asparagine. Its 3-letter designation is ASN, and its 1-letter designation is N, right? Not A. And I picked these particular two because valine here is nonpolar because, like I was saying, it has an alkyl group, carbons and hydrogens right there, right? Whereas asparagine is polar because it has this carbonyl group, remember? And in the carbonyl group, we have this C, right? And then the C group is Everything is partially positive over here at the carbon, right? And partially negative over there at the oxygen. So that would be a polar amino acid, right? And remember, the rest of it stays the same. Here's the N-terminal side, which is the amino acid side of the valine. The carboxylic acid side, or the C-terminal side of the of it. And then there's the hydrogen. Same thing here, right? There and there. So just remember that every amino acid has that same basic structure. Amine on the alpha carbon, carboxylate on the alpha carbon, and hydrogen on the alpha carbon, right? And then the R group, which is also on the alpha carbon, right? So talking about nonpolar and polar amino acids, that alludes us to the next set of slides. We have a number of nonpolar amino acids, right? And those are basically the ones that are that have mostly hydrocarbons in them, right? So the first nonpolar amino acid is glycine. Its R group is just a hydrogen, right? Its three-letter designation is GYL, GLY, and then its one-letter designation is just G, right? So that was pretty easy. Hydrogen as an R group, glycine, GLY, and then G. Next one, the methyl group, right? That's alanine. We saw that in the previous slide. Three-letter designation is ALA, and one-letter designation is just A for alanine, right? One-letter designation. We already saw valine on the previous slide. That's nonpolar, right? Because it has an alkyl group right here. Here's another alkyl group, a larger alkyl group, leucine. Its three-letter designation is LEU, and its one-letter designation is L, right? And it has got a CH2, CH3 on both ends right there, right? So basically, leucine is the same thing as valine, right? Leucine here is the same thing as valine, right? Except that it has an extra methyl in there, right? It has an extra methyl. And then we have isoleucine. Well, remember what iso means when you put it in front of a molecule's name. It means that it's the same molecule formula, molecular formula, right? It's just structurally different, right? So how is isoleucine's R group the same as leucine's, right? And how is it different? Well, for one thing, isoleucine has 3 carbons in a row, right? Right after the alpha carbon, there's 1, 2, 3. It's the same thing with leucine. 1, 2, 3. The difference between those two, though, is that the methyl group comes off from this carbon first. Comes from that carbon. The one that's right adjacent to the alpha carbon in isoleucine. Whereas in leucine, the methyl group comes off of the second carbon from the alpha carbon, right? So basically the same molecule formula-wise, but structurally only differing by the position of that one methyl group 1 carbon away, right? Right? Its 3-letter designation is ILE, and its 1-letter designation is L. Oh, sorry, not L, I, right? Leucine's is I. Yeah, we had to memorize all these. There's more, 20 total, when I was an undergrad. But I don't think I'm going to make you memorize those structures, so you should be fine. But just know, the properties and know the structures and what they mean and all that stuff. But you don't actually have to memorize. Like I'm never going to ask you to draw out one of these things, right? Just from memory. Let's see here. Next, we have phenylalanine, which is actually my favorite amino acid. I know that's kind of nerdy to have a favorite amino acid, right? But this is my favorite. If I ever have a bonus question and I ask you what's my favorite amino acid, it's phenylalanine. Same core right here, right? The amino, alpha carbon, carboxylate with the hydrogen, and the R group is got a CH2 and a benzene ring, right? Well, why do you think they call it phenylalanine then, right? What's another word for benzene ring? It's phenyl. Remember that from when we were talking about hydrocarbons? Phenyl, right? And then, what is it when you have just the methyl group right there off the alpha carbon? Well, it's called an alanine, right? Phenylalanine. Let me just tell you that biochemists are really simple, right? We we tend to name things pretty much self-explanatorily, right? And you'll even find later on when we get to the enzymes, we literally name enzymes based on what they do, right? So one of the enzymes I'll talk about a little bit later is called luciferase that I studied back in grad school. And luciferase, Lucy is actually Greek for the word light, right? And so race means enzyme, or sorry, not race, but ACE, A-C-E. So it acts on a molecule called luciferin, right? So that's why we call it luciferase, because luciferin is a light-generating molecule, right? That's why it's called luciferin. And this enzyme acts on that molecule to make it make light, and so we call it luciferase. So you'll see a pattern in which the substrate for the enzyme is always the name of the molecule itself, and then the enzyme that acts on that substrate is its name plus ACE at the end, right? We're that simple as biochemists, and you'll see that in a minute. Next, we have a methionine. Methionine has two methyls, right there, plus a sulfur, and then another methyl at the end. That's methionine. Oh, I forgot to say, the three-letter designation for phenylalanine is PHE, and you would think it would be P for the first letter, for one-letter designation, but it's not. It's F. F is the one-letter designation for phenylalanine, right? Let's jump to methionine now. Methionine again, two methyl groups to sulfur to another methyl group, right? And that's methionine. It's got MET as its three-letter designation, and M as its single-letter designation or one-letter designation. Methionine is very special, so we'll talk about this a little bit more in nucleic acids, but nucleic acids are basically DNA, right? And a DNA sequence is composed of codons, and those codons are basically a series of 3 DNA molecules at a time, right? Or 3 nitrogenous bases at nucleic acids at a time, right? They correspond to 3 other ones called anticodons, right? And then those 3 anticodons attach to an amino acid. And so when you read a DNA molecule, like a strand of DNA, you read them 3 at a time, right? And every 3 will code for an amino acid. Well, methionine, right, its codon, it actually codes for start, which means that if you have a methionine at the very beginning as the coded amino acid for a DNA strand, what that means is that you need to start making a protein, right? That's a start protein, right? There's also an end protein. There's also an end end sequence too. It doesn't have an amino acid attached to it, but it's an end sequence. I believe it's. I'm not a geneticist, but I'm pretty sure it's TGA, right? We'll talk more about that later, but I just want you to know that methionine is special. It's known as the stop codon. Known as the start codon, right? Start codon. Next, we have proline. Proline is special too. Its 3-letter designation is pro, P-R-O. 1-letter designation is P. But it's special because it's the only amino acid that has its alkyl group come back around and reattach to the amine group, making this 5-membered ring, right? So that makes it really rigid, right? It makes it really rigid. It doesn't get to spin as much. The rest of these guys, they're all on single bonds, right, to the alpha carbon. All these R groups are on alpha bonds. All these R groups are on sigma bonds, right, to the alpha carbon, right? And so, if you remember from when we were talking about the stuff in the hydrocarbon section, when you have 2 atoms around a sigma bond, right, or a single bond. I'm sorry, I'm saying sigma, but I meant single. Around a single bond, they spin freely. So all the R groups up until proline, they were all on single bonds to the alpha carbon, and so they spin freely. They have a lot of freedom. Proline, though, since it wraps around, its alkyl group wraps around and reattaches is right there at the amine, right? It's very rigid. It doesn't spin freely. It doesn't spin very freely, right? And then we have tryptophan. This is the same tryptophan that everybody thinks makes you sleepy when you eat turkey, right? And its R group is the methyl, of course, again in between, and then we have a benzene ring with a 5-membered ring, right, with a hydrogen. All right, so that's tryptophan. Tryptophan's 3-letter designation, CRP, and this is hard to remember. Its single-letter designation is W. I promise the only reason why it's like that is because T was already used for something else. That's why. All right, let's keep on going. We now have the polar amino acids, right? Here's serine. Serine's actually very special. Serine is always a part of things called the active site of an enzyme, and we'll talk more about that in the enzyme lecture next, right? But know that serine's important. Serine's R group is a methyl plus an OH, and if it's an OH, right, the oxygen makes it super polar. Really easy 3-letter designation. SER, really easy single-letter designation, right? I'm not gonna make you like actually draw out any of these, like on a test, draw out any of these amino acids or something, but you'll still have to be able to tell me what the 3-letter designations are and the 1-letter designations are, or maybe see these amino acid structures and kind of know what they are, right? But you don't have to actually draw them out, but from memory. Next, that's where the T went. That's threonine. Threonine is the same thing, right, as serine, except it has a CH3 methyl group coming off of that beta carbon, right, or that second carbon from the alpha carbon. Then we have tyrosine, right, another polar one because of this OH up here. It has a benzene ring in between the OH and the little methyl group right there, tyrosine, TYR, and Y as its single letter designation. We have cysteine here. Cysteine is very important too. Cysteine forms these things called disulfide bonds. They're one of the covalent interactions that holds proteins together, right? Two of them can get together and oxidize and become a disulfide bond, which we'll talk about together. We'll talk about in a second, right? And that will hold proteins together pretty permanently. And so that's CH2 with an SH. So remember I was talking about the father of enzyme kinetics, Mo Cleland, right? He actually was the very first scientist to ever use a molecule called dithiothiotol. It's actually DTT. It's commonly known as Cleland's reagent, right? And he was the first one to use DTT, right, to reduce these disulfide bonds right here, right, to separate cysteines in a protein, right, or an enzyme. So he has that name, Cleland's reagent. He was able to use that DTT, and so now he's got a chemical, a very important chemical named after him, Cleland's reagent. And I'm a part of that lineage, right? Because Cleland was my boss's boss, and then my boss is my boss, right? So, yeah, that was pretty cool. Sadly, though, Mo Cleland died a few years ago. We found out, but it's very sad. Anyway, he died while I was still in grad school, so I guess it was more than a few years ago. It was before 2016, somewhere between 2012 and 2016. I can't remember the exact date. Anyway, next one is asparagine, right? Asparagine is got the amide, right, along with the CH2, and then we have glutamine, right, which also has the amide with the CH2, but it has an extra CH2, right, making it a little bit longer. All these are polar, of course, right? All these are polar. And so, if you remember, we had asparagine in the previous slide, and ASN and N are its designations, and then for glutamine, GLN and Q for its designations. Wow, Q, that's hard. Q for glutamine. Right? But just remember though, if you're having trouble remembering it, asparagine and glutamine are basically the same thing, except that glutamine has one extra carbon or one extra CH2, right? Serine and threonine, right, and tyrosine and cysteine are all the same, right? From there down, okay? Just that serine has an OH. Threonine has a CH3 extra and an OH. The tyrosine has a phenol group. I don't think I ever told you that before. Whenever you have a phenyl with an OH, it's called a phenol, right? And then cysteine has the SH. So you can start to glean the fact that I've used the word glean twice now. That's weird in one lecture. Anyway, you can glean the fact that and some insight into that that amino acids are very similar to each other. They just have slightly different R groups, right? Even past the beta carbon, right? And that's how I memorize them, right? Let's keep going. We have acidic amino acids too, right? The acidic amino acids, or aspartate, sorry, lost my train of thought for a second there, are aspartate and glutamate, right? Now, what does that sound like? That sounds a lot like the previous slide where we had, previous, there we go, where we had asparagine and glutamine, right? Where asparagine and glutamine had an amide there, right? Aspartate and glutamate are exactly the same thing, except instead of having an amide, they have carboxylic acids there. Specifically, it's a carboxylate, right? It's not a carboxylic acid, but it's a carboxylate ion, right? Those are the acidic amino acids. Why is the carboxylate? Just a quick review, because there's no hydrogen there. There's just a minus sign. Makes it a carboxylate on both. Aspartate, ASP, and D. Glutamate, GLU, GLU, and E, right? Now you know why the other ones didn't have regular 3-letter designations that were based on their name. It's close though. Next, we have the basic amino acids. We have histidine. Histidine's a very important one. We used to do this thing, or we still do this thing, where we put a 6-histidine tag on any protein that we're trying to make, or enzyme we're trying to synthesize, and we can use that 6-histidine protein, or 6-histidine in a row, 6 histidines in a row to purify that protein or enzyme from the cell lysate, right? Because this is very very rare that you have 6 of these in a row right here, 6 histidines. And histidines are a nitrogen-based 5-member ring on a CH2, right? With the positive charge on that nitrogen and 2 hydrogens, right? That's histidine. That's histidine. Okay. That's histidine. Let me clear that real quick because I got a little messy. There we go. There it goes. Positive charge on that nitrogen. Okay. So what does that actually mean then? At physiological pH, this hydrogen can actually go back and forth from being there, and that's why there's a positive charge right there. And so sometimes you'll see histidine without this hydrogen on there, and that plus sign is gone. Because the plus sign is only there when there's 4 bonds to nitrogen. Right? Easy to remember. His as a 3-letter designation. H for the 1-letter designation. Next, leucine. Super long alkyl chain, right, from the alpha carbon. And then a ammonium at the end. Alright? With ammonium at the end. Lysine, LYS, and K. And the arginine, just a tiny bit more complicated. Another long alkyl chain, and then this NH group here, and this NH group here, with this carbon in the center, and the acidic basic part there, right? Acidic slash basic part there. Arginine, Arg, it's the 3-letter designation. Arg, maybe, Arg, that's better. Arg, like a pirate, right? And then R is its single letter designation, right? And those are the four different, groups of amino acids. So expect questions on the exam that are like, is this a polar amino acid? Is this a acidic amino acid? Is this a non-polar amino acid? Those kinds of things. I've already gone over this before, but just a quick review. We have aspartic acid here, or aspartate here, and it's not aspartic acid; it's aspartate because it's a carboxylate, right? But anyway, I've already gone over this, right? This part here is the R group, and then every single amino acid has the NH3 plus to the alpha carbon, H to the carboxylate, right? That might not look like a carboxylate, but that's the same thing as doing this, right? And that may not look like an amine over there, an ammonium right there, but it could also be this. You can also draw it this way, right? Yep. Same thing here. Look, you see the asparagine, right? I already said that before. Same thing as aspartate, except it's an amide instead of a carboxylate, right? So when you're drawing amino acids, not to say that you will have to draw amino acids, but if you ever draw an amino acid, just remember you have to have this part first. That is the most important part of the amino. Well, I'm sorry, it's not the most important part, but it's the backbone of the amino acid. And then the R group is the important part of the amino acid because that's the business end. That's what does things. Some don't do anything at all, actually. So, amino acid practice. We're drawing valine. I actually like to remember. I remember valine because valine, V. I think, I think that the biochemists draw valine or they saw the molecule valine, and they were like, oh look, it makes the shape of a V. That must be what valine is. Let's just name it valine. So it's pretty simple to draw valine. Always start out with the backbone, right, or the alpha carbon part, and then draw the rest of it, right? And that's valine. Well, I messed up a little there because there should be carbons there. I went from expanded formula to line angle there. That's bad. Let me do that again for you. Bad habits as a biochemist. Sorry. There we go. Bad habits. Draw them really fast. Right, with that, and then CH2. CH3, CH3, all right? And this is at physiological pH. This is what is going to be a physiological pH. Positive charge with an extra hydrogen on the amino group, ammonium group, and then negative charge without the without the hydrogen on the carboxylate, right? We're at the three-letter designation, easy. VAL for valine, and luckily for us, valine is the only one that's V, so it has the V as its single letter designation. Next, we have cysteine, another simple one. NH3, whoops, H3. If I can get this N out, there we go, and then the rest of the alpha carbon part of the amino acid, right? And it's just the CH2 with the OH. Oops, not OH. I did that wrong. Look, see, even I make mistakes. It's SH. Oh man, Moclidon would be so upset with me for that because that's what he uses to do disulfide bonds, or at least reduces these disulfide bonds, right? Right. So CYS is the cyst. No, there we go. CYS is the three-letter designation, and I think C is the single-letter designation. Let me think about that for a second. Yes, I think that is C. I'll come back and fix this if it's not. Anyway, let's keep going. So, I've been talking about this a lot, and I've never really showed you what it was. Peptides, right? What are peptides? Well, peptides are a string of amino acids that are linked together by what we call a peptide bond, right? So here we have a glycine and an alanine right there. This is the glycine right here, right? And this is the alanine right here. Okay? And it's being held together by this bond right here. That's the amide bond, right? Well, it's a peptide bond, but it's also an amide bond, right? So a peptide bond is always also known as an amide bond, right? Amide bond. Again, linking two or more amino acids together by peptide/amide bond is makes a peptide, and you can imagine how long these things can get, right? Gazillion amino acids together, right? So I've been talking about peptides a lot in the last few slides, and I've never actually told you what they are. What are peptides? Well, peptides are basically amide bonds, right? What is an amide bond? It's that bond that's between the carbon and the nitrogen of an amide group, the amide functional group, right? So peptide bonds are amide bonds, right? They're formed between the carboxylate of one amino acid and the amine of the other amino acid, the next one down, right? The next one to it. And like I was saying before already, linking two of those or more amino acids by a peptide bond forms a peptide, okay? So a peptide is basically a series of amino acids that are linked together, one of the many of the 20 all linked together, right, by an amide bond, and you make a peptide. So I will use probably amide and peptides interchangeably, so just know that. They are the same type of bond. So looking over at this example over here, we've got a glycine over here, right? This side is glycine. And then over here, we have an alanine. We know it's an alanine because there's the methyl group of the R group right there, right? And then there's over here we know it's a glycine because there's the hydrogen of the R group right there, right? And then they're bonded together, forming what we call a dipeptide. A dipeptide, right? Using a amide bond right there, or a peptide bond, right? And this entire group right here, of course, is an amide functional group. So when you form a peptide bond, the two amino acids on either side of the amide functional group, right, are bonded together by the peptide bond, right? And then the thing that you need to glean, oh my god, that's like the 5th time I've used the word glean today. You can glean from this that once you get more amino acids attaching to it, right, with the peptide bonds, and it becomes a peptide, that sequence of amino acids is known as the primary structure of a protein, right? It's known as the primary structure of a protein. We'll go through the different types of structures of proteins, but the first one you need to know is the primary structure, and that is quite literally the peptide bond, or the peptide, right, which is the amino acids that are in that sequence, right, that are attached to each other by a peptide bond, right? That's the most basic structure of a protein, the primary structure. And this is just an easier way of seeing it. We have an expanded structural formula right here. There's the peptide bond, right? Also known as an amide bond right there. That is between the glycine over here and the alanine on that side. And then as I mentioned earlier, the side with the amino group is called the N-terminus, or the N-terminal and, excuse me. And the side with this carboxylate is the C-terminus, or the C-terminal side. I've heard people use it either way. Terminus, terminal, all means it's the end, right? So let's keep on trucking along here. All right, let's do some peptide practice. So the question says to draw a dipeptide of serine and threonine, right? Remember that remember that serine is the one with the R group that had the CH2, right? OH, right? That's its R group. And then threonine was almost exactly the same thing. CH2, actually it's CH, right? Except it had an extra methyl group at the end, right? So that's serine, and that's threonine. And so when I do this, I always draw out the R group so I can remember what they look like, because even I forget them sometimes, and I'm supposedly a formerly trained biochemist. Anyway, and then once I get that straight, and I know what the R groups are, I draw the backbones first. And so I start out by drawing the amine group, NH3+, right? And then the alpha carbon down to the carboxylate, and then up to the next amine group, right, to the alpha carbon, and then back to the carboxylate, right? And so remember that this part right here, right there, that's the peptide bond. Right? That's the peptide bond. This side right here is the C terminus. And this side right here is the N terminus. Right? And I've drawn everything there except for the hydrogens and the R groups on the alpha carbons, right? So since the amino groups and the carboxylate is pointing downwards on the serine, I'll draw the hydrogen up there like that, and the R group, CH2OH, up there like that, right? And then similarly here, since the amine is pointing upwards this time, and so is the carboxylate, I'll make the hydrogen pointing down with threonine's R group coming downwards. And that's how you draw the dipeptide serine threonine, right? Serine threonine. The important part is just to remember, right, that this part right here is the peptide bond. That's how you join two amino acids together. And if you look at it closely, it just looks like an amide bond. So an amide bond is basically, a peptide bond is a amide bond that's inside of a peptide, right? They're the same thing. Let's keep on moving. So I alluded to this before, right? The primary structure of proteins is actually the amino acid, right? Sequence, right? It's the amino acids. So when you look at this one, for example, we start out with alanine, glycine, and serine, right? Put it all together and you get alinoglycylserine, which is its name, the name of the peptide, right? And the primary structure is literally the sequence of alanine, glycine, and serine together, right? Here's another amino acid sequence that's also known as the primary structure, right? The primary structure of this peptide is alanine, right? So we know that because of the methyl group right there. Leucine, we know that because of this alkyl group right here. Then cysteine, SH right there with the CH2. And then methionine, right? That's how we know. So when we say primary structure, just remember that the primary structure of a protein is the actual, here it says particular sequence of amino acids that are held together by the peptide bonds. And of course, if we're talking about primary structures, there are going to be secondaries, tertiaries, and quaternaries, but we'll get to that in a sec. So in the previous slide, if you recall, I was talking about how proteins can function as hormones, right? Hormones are things that trigger certain biological functions, right? And the peptide of sequence with primary sequence glutamate, histidine, and proline is actually a thyroid hormone, right? And that thyroid hormone activates or stimulates the release of thyroxine, right? In your body. A more interesting one is insulin. Insulin is also a peptide hormone, right? It's got two chains in it, an A chain and a B chain, right? And the A chain has 21 amino acids in it, and the B chain has 30 amino acids in it, right? And you can see here that they're being held together by these things here. And I was telling you about these before because my boss's boss was the one that used dithiothiotol to reduce those. But these are the disulfide bonds. Disulfide bonds, right? They're the covalent interaction that holds proteins together, or in this case, primary structures together, right? And you notice here there's a sequence of amino acids that are going through from the N terminal to the C terminal, right, on both chain A and chain B. And it's that sequence of amino acids that makes insulin do what it does, right? Because there are receptors on your liver cells and on your muscles, and that will accept the insulin, right, and trigger the storage of glucose as glycogen, right? Which I think we talked about a little bit in the sugar lecture, right? So peptides are very important, right? And the important thing about these particular peptides is that they are a sequence of amino acids that are primary structure, but they don't go into secondary or tertiary or quaternary structures to be functional, right? We'll talk about those in a second. Moving along, we have some more important peptides. We have oxytocin here. It stimulates the uterine contractions in labor, right? It's got this amino acid sequence right here, right? Cysteine, threonine, phenylalanine, isoleucine. Sorry. Oxytocin has isoleucine. Vasopressin has phenylalanine there, right? Glutamine, asparagine, cysteine, proline. Same thing here. Oxytocin has leucine there, and vasopressin has arginine there, and glycine. And they're being this primary structure is being held together covalently with another disulfide bond, right? Another disulfide bond. So interestingly enough, though, just a change in amino acids at one position, or in this case, two positions, right, turns oxytocin into vasopressin, right? Vasopressin, the antidiuretic hormone that regulates blood pressure, right? So you can see how just changing single amino acids, 2 or 3 amino acids, or even deleting an amino acid or adding an amino acid can change the function of proteins and enzymes, right? Some leading to disease, right? Here's the amino acid or primary sequence of beta endorphin, right? All these like well-known like hormones and drugs and such, many of them are peptide sequences, or are short peptides primary sequences, right? Moving on. Oh, more peptide practice. How exciting. Okay, amino acid. What amino acid is at the N-terminus? Well, the amino acid at the N-terminus is this guy right here, right? What is that one? Anybody remember? I'm giving you 5 guesses. Did you guys guess? I don't know if you did or not, but I'll go ahead and tell you. The N-terminal amino acid is phenylalanine. Right? You know that because remember, an alanine has just the CH3 there, or methyl group there, and then you add it on a phenyl group. Right? So that's phenylalanine. Phenylalanine. What is the amino acid at the C terminus? Right? Well, that's this guy right here. That's that guy right there. And what's up there? Dang, I already gave it away in the last one. When it's just a methyl group, what is that? That's just an alanine. So what's the name of this tripeptide? Well, phenylalanine. And this is what? Anybody know what the middle one is? It's the one that my old boss's boss, you know? That whole thing I keep on telling you guys about? Yeah. Cysteine. I always spell it wrong. I think it's like that. Yeah, I think that's right. I before E, except after C. Yeah. So phenylalanine cysteine alanyl. I forgot to add in the neals at the end. So let's try that again. Phenylalanine alanyl, right? Cysteine alanine. I don't think I'll ever ask you to do that, but just be able to identify these amino acids, right? Phenylalanine cysteine alanine. That's the name of that that tripeptide, right? Moving forward, after we have a primary structure, we have this thing called a secondary structure. There are multiple secondary structures. One of the most common, though, is called the alpha helix. Very much like the double helix of DNA, except this is only a single helix, right? We'll call it an alpha helix. Okay, and the way it forms is that a hydrogen bond forms between the carbonyl of the carbonyl group, right, and the hydrogen, the oxygen of the carbonyl group, and the hydrogen of the amine group, right, of the amide bonds in the next turn of the alpha helix, right? So literally, what's happening here is that we have an amino acid, right, that's right here on the N-terminal end, and it's bending around, and it has a hydrogen right there, right? And that hydrogen is bonding with the oxygen from the carbonyl of the next peptide bond, right? It's interesting because this goes all the way up the chain. You can see it here. There's one there, right, that I just pointed out to you. There's one there. There's one there. This one there, and one there, and one there, and one there, right? And the interesting thing about it is that this happens throughout the entire peptide chain, and if it's a certain type of amino acid, right, that allows for very close quarters like that, right, where its steric hindrance is minimal enough that it can get close enough to do that hydrogen bonding, you'll get the amino acid, the amino acid chain or the peptide chain to spiral, right, spiral around like an alpha helix, right? It's pretty neat. It's pretty neat. So that's the alpha helix, right? That's the alpha helix. If we keep on going, another secondary structure is known as the beta-pleated sheet, right? And on the beta-pleated sheet, same kind of thing going on here, where we have hydrogen bonds occurring between the hydrogen of the amide and the oxygen of the carbonyl of amino acids, right? And if the R groups are small enough, and in this case, if they're, for example, methyls or just hydrogens, they can get even closer to each other. And when they get this close to each other, right? When they get this close to each other, you can form a beta pleated sheet, and it really looks like just like a sheet of paper that's kind of ruffled, right? It looks like a sheet of paper that's kind of ruffled, three-dimensionally, which is pretty neat. Pretty neat. All right, moving on. Another one known as the triple helix is basically like an alpha helix with three of them wrapped with each other, right? Three peptide chains wrapped with each other. Excuse me. And so those are typical in things like collagen, connective tissue, skin, tendons, and cartilage, right? Collagen fibers are triple helices of peptide chains held together by hydrogen bonds, which you already know that because alpha helices are held together by hydrogen bonds too. So in this case, not only do you have the hydrogen bonds holding the chain, the single chain together to make the spiral, like just within the chain, but then with the other two chains, there are hydrogen bonding occurring too, that holding those together too. And you've got to have some really small, like R groups, to be able to overcome the steric hindrance to make a triple helix that's woven that tightly together, right? But all these, excuse me, all these structures, these alpha helices, the beta sheets, beta pleated sheets, the triple helix, they're all being held together by hydrogen bonding. Just remember that. Right? That's what I want you to remember from that. So, turns out that there are a lot of diseases that have to do with secondary structures. And one that is very prevalent is Alzheimer's disease. Alzheimer's disease. I've never been able to say that right. Signs of dementia, increasing memory loss, inability to handle daily operations. Those are the things you look out for when it comes to Alzheimer's disease, right? And what's actually happening here with Alzheimer's, which there's not a cure to yet, right? But in a person's brain with Alzheimer's, the beta amyloid proteins change shape from a normal alpha helix that are soluble to sticky beta pleated sheets, right? So here's what it's supposed to be inside the brain that are soluble, and then they turn these into these sticky beta pleated sheets, right? And the problem with that is that these beta pleated sheets are the fragments that become the plaques that are prevalent in the neurons, right, of an Alzheimer's brain, right? The diagnosis of Alzheimer's disease is based on the presence of plaque and neuron neurofibrillary tangles. That's a hard word for me for some reason. In the neurons that affect the transmission of nerve cells, right? So when you have those beta pleated sheets that are not soluble, right? And you have the neurofibrillary tangles, they inhibit the transmission of nerve signals, and so that's that's what Alzheimer's is about, right? Sadly, there's not a cure for Alzheimer's disease, but if you're ever wondering, that's how Alzheimer's happens, right? These alpha helices that are normally water soluble, right? They become these beta pleated sheets that are insoluble and sticky, right? Next comes the tertiary structures, and these are what we all, as biochemists, when we say protein or an enzyme, right? We think of them in these types of structures, tertiary structures, right? So, in a tertiary structure, it's the actual three-dimensional structure of a protein that includes the beta pleated sheets and the alpha helices, right? And everything that's being held together with the covalent bond, the disulfide bond, right? But in a tertiary structure, the protein is being held together by the non-covalent interactions inside the protein, right? Non-covalent interactions between the different side chains of or R groups of the amino acids, not the covalent interaction, right? The covalent interaction was the disulfide bond I was talking about before. So these non-covalent interactions, right, are the hydrophobic interactions, right, between two nonpolar amino acids, right? Hydrophilic interactions between the external aqueous environment and the R groups of the polar amino acids. We have salt bridges, which are ionic bonds between the ionized groups of basic and acidic amino acids. And then we have the hydrogen bonding, which we talked about before, between the amino acids that form alpha helices and beta plated sheets, right? And I already spoke about this before, the disulfide bonds, which are the one covalent interaction that holds three-dimensional structures of protein together. So let's first talk about the hydrophobic interactions. It's exactly what it sounds like. It's the interactions between nonpolar molecules because they're afraid of water. That's what hydrophobic comes from, right? And they tend to want to be together. Those hydrophobic molecules want to be together, right? So they can minimize their energy and keep the water on the outside, right? So that's what a hydrophobic interaction is. Obviously, hydrophobic interactions occur with the nonpolar amino acids. Things like phenylalanine or glycine, alanine, those kinds of things, right? So let me see if I can find a hydrophilic interaction somewhere here. Hydrophobic interaction, right here. Right here, there's a hydrophobic interaction right there between two phenylalanine groups, right? And notice, right, here's another hydrophobic interaction over here, two alanines, right? And it's actively keeping the water molecules outside of that area, right? Making the area where it's all combined or all close to each other hydrophobic, right? So as far as hydrophobic interactions go, we have here the tyrosine and serine, right? Oh, I'm sorry, I said hydrophobic. I meant hydrophilic. The hydrophilic interactions go. We have the tyrosine and serine here, right? And they both have OH groups, right? Very similar to water having its H2O, and that's why it's a hydrophilic interaction, right? And so it's perfectly happy being outside, being exposed to this environment of water, right? This is all water out here, right? Right? All water outside. Then we have salt bridges, right? Again, so some examples. Here's a salt bridge right here between the carboxylate of that amino acid and the amine group of that amino acid. That's a salt bridge too. Salt bridges are also they like water, so it's not a big deal there. But those are the salt bridges. You know they're salt bridges because they have charges right there, plus sign, minus sign, right? Are there any other salt bridges? I think that was the only one. And then we have hydrogen bonding. There's hydrogen bonding all throughout this molecule, right? There's a bunch of hydrogen bonds happening in there, in the alpha helix there. There's a bunch of hydrogen bonding happening there, in the beta pleated sheet there. All these alpha helix have hydrogen bonding, but it does point out one specific hydrogen bond right here between these two serines right here, right? These two serines of two separate helices, right? Two separate helices. That's hydrogen bonding. And then, last but not least, an example of disulfide bonds right here, right? And they're holding together covalently, right? That's why you see that when you look at a disulfide bond, it's a solid line between the two S's. But when you look at things like hydrogen bonding, there's just three dots, right? Or hydrophobic interaction, doesn't look like there's a dots or lines at all. Hydrophobic or hydrophilic, right? Well, that's because those are all non-covalent. Same thing with the salt bridge, right? I accidentally marked out the plus sign that was on that nitrogen right there, but it's a nitrogen plus ammonium group right there, right? But yeah, those are the examples of the four non-covalent interactions, right? I like to call these the four non-covalent interactions. Non-covalent interactions. And then the one covalent interaction. Covalent interaction that holds the tertiary structure of proteins and amino acids together. Right. Moving forward, we have the quaternary structure. Right. This should look very familiar to you. This is the exact same example I gave you on the very first day of class about hemoglobin. The quaternary structure of a protein is, oh, that's really weird. There's a font problem right there. That should be a bigger font. Not like a material problem, right, but a font problem. Anyway, so quaternary structures are basically a bunch of tertiary structures being held together, right? And so if you look at, for example, the hemoglobin here, I forgot to say that. Hemoglobin, I always forget if hemoglobin has an A or not in it. It doesn't have an A. Hemoglobin, right? That's hemoglobin. And if you look at hemoglobin, it looks like it's made up of multiple tertiary structures, right? Multiple. The two red chains are alpha chains, right? Which are this guy and this guy, right? And then the two interesting, that's a typo. Oh, I got a little confused here because it said two blue are the beta chains. Where are the beta chains at? Because this is orange. Maybe I used the wrong picture. Anyway, 4 subunits, right? 4 subunits. And the one thing that is correct here is the heme group. And there's the heme group right there, and it's in green, right? Multiple heme groups. But the point of this particular picture is that hemoglobin is made up of 4, 2 plus 2, right? 4 subunits, right? And there are, and they all 4 of them bind oxygen, right? There's a heme there that binds oxygen. There's a heme group there that binds oxygen. There's a heme group there that binds oxygen. And there's a heme group there that binds oxygen. Yeah, so, yeah. That's very confusing. I feel like this is the wrong picture, but it doesn't matter because 2 of them are, 2 of one color are the alpha subunit, and 2 of the other color are the beta subunits, or beta chains, right? And whoever, me, wrote that there as blue and red, I'm obviously colorblind. No, I'm not colorblind. That was just a typo, I guess. Anyway, moving forward. But that's a quaternary structure. So, just as a quick review, and this is the part I really want you to know, the primary structure, right, goes to the secondary structure, goes to the tertiary structure, goes to the quaternary structure, right? Yeah, see, I have the same picture here again, so I must have just picked the wrong colors on the last slide. Anyway, yes, that's the order. Primary, excuse me, which is the amino acid sequence, right? Remember that. Amino acid sequence is the primary structure. And then the secondary structure, right here, is the alpha helix. Let me make an alpha, like a true Greek person there. There we go. The alpha helix. Which could actually be a beta-pleated sheet too, right? These form with hydrogen bonding. Right. And then from there, the secondary structure can become a tertiary structure when a whole bunch of them get together and form the 4 non-covalent interactions to hold together, right? And the 1 covalent interaction. What were those 4 again? Hydrophobic interactions. Hydrophilic. Right? Then salt bridges. Or we can also call those ionic interactions. And then last but not least, in the non-covalent, hydrogen bonding. And then the one covalent interaction is what? The disulfide bond. There we go. So, sorry that took a second there. My finger accidentally went onto the Siri button. And Siri kept on coming up. Anyway, keep going. The show must move forward. Next. So, just a quick summary of everything, which I just did a second ago, which tells you how important this stuff is, right? At the primary level of protein structure, we have the amino acids that join, becoming a specific polypeptide sequence, right? At the secondary structure level, we have the alpha helices and the beta sheets that are formed by hydrogen bonding between the atoms of the peptide backbone. At the tertiary, we have the three-dimensional structure of the polypeptides as they fold into compact three-dimensional shapes that are stabilized by the non-covalent interactions like the hydrogen bonds, salt bridges, hydrophobic interactions, and the hydrophilic interactions, and the covalent bond interaction, which is the disulfide bond, right? This is important. I didn't even say this before. This is what forms the biologically active protein. That's important. Okay. And the quaternary structures are basically two or more protein units, right, that are tertiary structures that are stabilized. They combine together and stabilize with the same non-covalent and covalent interactions and become an active biological protein, right? So over here to the right, I actually have some interesting stuff. When I was in grad school, I studied two different enzymes. I already told you about one before. The first one was luciferase, and that's this guy right here. This is luciferase. So what luciferase does is that it's an enzyme that catalyzes a reaction with the substrate luciferin, and it will, in this reaction, produce blue light, right? And I thought, oh man, that's really cool. I'm going to study that, figure out how that works, right? So this is actually luciferin here, and you can see in luciferin, it actually has a beta-pleated sheet right here, right? But its interactions are so interesting that the beta-pleated sheet will wraps around itself, right, making what we call a beta barrel, right? So the sheet folds around, it becomes a cylinder, and we call that a beta barrel. The other interesting thing too is all the alpha helices you see, but specifically these three alpha helices up here with those H's there. The H stands for histidine, and the number is the number in the sequence of where the histidine is located in the primary structure, right? That alpha helix those alpha helices act like a little lid to the beta barrel, right? So here's the barrel, right? And here's the three alpha helices, right? And based on pH, those alpha helices / those histidines, those three that are on the picture right there, right, will either open or close the beta barrel. And if the beta barrel is open, luciferin, the substrate, can go inside the beta barrel, right? And when it goes inside the beta barrel, the luciferase will act on it and produce blue light, right? But if the beta barrel is closed, then luciferin can't go in, and it won't make any blue light, right? And it's all based on pH. It's actually really cool. Then up here, we have an enzyme called MCR. Notice that it has multiple subunits. Oh, you know what? That's probably where I got the blue from. I was probably looking at that, like blue versus red. Anyway, we all make mistakes, right? So, it has multiple subunits, and this is called MCR. MCR is short for methyl coenzyme M reductase, right? And what methyl coenzyme M reductase does is that it can change things like CO2 into methane, right? Which is awesome because methane is a fuel and CO2 is a greenhouse gas, right? In a process called methanogenesis. Methanogenesis. So another protein disease is sickle cell anemia, right? And in sickle cell anemia, we have hemoglobin, and hemoglobin in the red blood cell, right, has the normal, has a beta chain in it that has the normal sequence: valine, histidine, leucine, threonine, proline, glutamate, and lysine, right? But in a patient that has sickle cell anemia, that beta chain has this glutamate switched out, the sixth one right here, switched out with a valine, right? It's switched out with a valine. That causes all kind of problems. First of all, the glutamate is a polar amino acid, right? And the valine that got switched out for is a nonpolar amino acid. And remember what I said before, like all kinds of diseases can come about just from the addition of an amino acid, the subtraction of an amino acid, or the substitution of an amino acid. And here, the change is a glutamate to a valine. Bad problems happen then, because it turns the hemoglobin in the red blood cell into a really weird shape, right? Mostly because since you have a nonpolar amino acid now, right, instead of a polar amino acid, those nonpolar amino acids will do hydrophobic interactions with other parts of the hemoglobin that were not supposed to be there to begin with, right? And so it turns it into this weird shape, and what that does is that it diminishes the ability of the hemoglobin to transport oxygen, right? But to boot, those hydrophobic interactions cause things like clogs in arteries and inflammation and pain and organ damage, because since they're doing the hydrophobic interactions, they tend to clump up. And if you remember what I was saying before about hydrophobic interactions, they tend to clump up with each other, right? So that they can all be together and they can have the water on the outside of them, right? And when they clump up together, they can be really sticky, and they can clog up arteries and cause blood clots and stuff, right? So, and capillaries. So sickle cell is not a fun thing to have. Not a fun thing to have at all. Interestingly, though, the first time I was actually introduced to sickle cell anemia was in the context of parasitology. And we were studying the parasite Plasmodium, which causes malaria. And it turns out that patients with sickle cell anemia were less likely to be infected by malaria and the Plasmodium parasite, because the Plasmodium parasite couldn't extract enough nutrients from the sickle cell of the patients of sickle cell anemia, right? So I guess it's a trade-off. Do you want to have sickle cell anemia or do you want to have malaria? I wouldn't want to have either, honestly, right? But that's sickle cell anemia. Moving forward. Oh, denaturization of proteins, right? So, in biochemistry, we do a lot of denaturing proteins because oftentimes, we need to study them by figuring out what their size are, their properties are, and we have to do a bunch of different things to make them so that they come out to their primary structures, break apart from their tertiary structures, and come out to the primary structures, right? And the main way of doing that is with heat, right? Because a lot of the non-covalent interactions I was talking about was hydrogen bonding, right? And so if you think about it, hydrogen bonding is the same thing that keeps water together, right? And when you boil water up to 100 degrees Celsius, right, or 212 degrees Fahrenheit, what you're doing is you're actually introducing enough energy into that water that it overcomes the energy in the hydrogen bonds and allows for the water to evaporate, right? In a sense, you're denaturing the water. Not really, you're not denaturing the water. You're just making it possible for the water to evaporate by disrupting those hydrogen bonds. Well, it's a similar thing with proteins. You can use heat to break apart the hydrogen bonds, right? And disrupt the hydrophobic interactions in proteins, right? To denature them, to make them flatten out into their substituent structures, right? Acids and bases can also do that too, right? Those will break the hydrogen bonds between the R groups and ionic bonds as well. They're salt bridges. Heavy metals can also interact with the disulfide bonds, right? And then agitation, like physical agitation, actually like physically messing with them, right? Will stretch peptide bonds until they break, right? And so a really good example of denaturing proteins is eggs, right? Which I'm not really sure. Oh, yeah, okay. For some reason, I thought that was a lunch pail, but that's really just a gas stove and a frying pan. Like, why does that person have fried eggs just in their lunch bag with nothing around it? That doesn't make any sense. Why did I pick this picture? Anyway, the egg whites of a protein, right, are denatured when you heat them up, right, when you eat them, right? And the active protein, the egg white looks like that. And then you add in some heat, and then you stir it around a little bit, and then you add in some salt, just all the stuff that you add here, right? Like I was talking about in these little bullet points. And then you got a denatured protein, which is a cooked egg white, which actually tastes really good and healthy for you, right? So you literally disrupted the tertiary structure of the egg white, right, by cooking it in a frying pan and not just having it inside of your lunch pail with nothing at all. I'm losing it today. So there's a little summary of denaturing processes that you can use to disrupt proteins, right? Heating it above 50 degrees can disrupt the hydrogen bonds, the hydrophobic interactions between the non-R groups, right? Non-polar R groups. Examples of that is cooking food and autoclaving surgical items. That's how you killed the bacteria, right? On like scalpels and hemostats and such. Interestingly, though, this is above 150 degrees Celsius right there, right? But when you're autoclaving, you're actually autoclaving up to 121 degrees Celsius. That's a lot. What? There it goes. I don't know why I put a 0 at the end of that. 121 degrees Celsius. That's super hot, right? So acids and bases, of course, they disrupt the hydrogen bonds because almost anything can. But then it also disrupts the hydrogen bonds between the salt bridges. The bonds between the salt bridges, right? That's like using lactic acid from bacteria, which can denature milk proteins, right? That's why your milk goes bad, right? And the preparation of yogurt and cheese. Organic compounds can also denature proteins. Of course, they disrupt the hydrophobic interactions, adding things like ethanol, isopropyl alcohol, right, to disinfect wounds and prepare for skin injections. That kills bacteria that way, right? Like when I had to do that with my hand there because my dog bite and I had stitches. That hurt a lot. Lots of prostaglandins being produced there, by the way. Heavy metals, right? Like heavy metals like silver, lead, and mercury, right? They actually interrupt the disulfide bonds, right, by forming ionic bonds, right? They do these like little metal complexes with them, and that's how it helps that, right? And examples of that would be like mercury and lead poisoning. And then physical agitation. That disrupts the hydrophobic bonds, hydrogen bonds by stretching the polypeptide chains until they break, right? Like whipping ice cream or making meringue from egg whites. It's all the same, right? Baking. I started watching a show on Netflix. It's awesome. The Great British Baking Show. God, it's a good show. You should watch that. There's a lot of egg white whipping in there, which means there's a lot of physical agitation of proteins in there, which means you should watch it because it's about your class. But if anything, it's just a fun class, right? And that actually ends today's lecture. I think these dates are correct. Due today, by the end of the day, which is April 13th, right, we have Blackboard assignments 13 or 14 are due, and then also quiz 12, and covering lecture 12 is due by the end of the day today. So being assigned today and being due next week on April 20th will be the Blackboard assignments 15 and 16, and the quizzes 13 and 14, right? And that ends our lecture on the introduction to proteins. Our next lecture will be about enzymes, right? And I'll see you for that one in a few minutes.